Okay, previously we learned that the motor cortex, specifically primary motor cortex, can direct contraction of specific muscles if you use brief periods of stimulation within different parts of cortex. And we also learned that there's a map of the body, a somatotopic map, along primary motor cortex, where each individual part of that motor strip controls different parts of the body. We also learned that primary motor cortex can direct a combination of contractions to produce a specified outcome, usually when you stimulate a certain part of cortex for a longer period of time, say half a second. But there are other areas around primary motor cortex that also contribute to movement. For example, posterior parietal cortex, you'll remember that this is sort of the end of the dorsal stream in visual processing, and hopefully you'll remember that the dorsal stream is important for integrating vision and action. It's also called the Ware pathway. So neurons here respond to visual or somatosensory, that is touch stimuli. They can respond to current or future movements, and they can also sometimes respond to complicated combinations of a stimulus, let's say something you see in the visual world, and an upcoming response that you're planning toward that stimulus. When you damage this area, it often results in difficulty coordinating visual stimuli with movement, difficulty in integrating what you see with what you do. The premotor cortex also plays an important role in preparing for movement. It's generally active in preparation for a movement, a period of time before you actually initiate the movement. And it seems to encode information about the location of the target in space, the target being the thing that you're moving toward or acting on. It also seems to integrate information about position and posture of the body and organizes the direction of the movement in space. Mirror neurons are a relatively recent discovery in neuroscience. They were identified first in monkeys in their frontal cortex. We've since been able to identify areas where we think they exist in human brain as well. These discharge or become active both when the monkey does a particular action and when it observes other individuals, monkeys or humans, doing the same or a similar action, hence the mirror part of the name. So for example, in one of the earliest studies on this, they discovered a neuron that responded when monkeys picked up, let's say, a peanut. So as they reached down and picked up a peanut, this neuron would become active every time the monkey did that. The same neuron would also become active when the human experimenter reached down and picked up the peanut. There's been a lot made of mirror neurons. Psychologists have really grabbed onto this idea and have used the existence of mirror neurons as an explanation for all sorts of behaviors, everything from the ability to mimic what other people are doing all the way to our ability to empathize with other people. It's not clear to what degree mirror neurons are really crucial for any of these functions, but really understanding their role in behavior is going to require some more work. Here's an interesting study that suggests that these neurons aren't just there for helping understand actions, as has been proposed, but they may be important for understanding the intentions of actions as well. I'm going to briefly walk you through this study conducted in 2004 by Giacomo Rizzolatti and his colleagues. Rizzolatti and his colleagues were the ones who initially discovered mirror neurons. So here they're recording from an individual neuron that seemed to respond when a monkey reached out and grabbed something, or when it observed a hand reaching out and grabbing something, like this block. Each one of these rows here is one trial, one instance of the monkey observing this little video of a hand reaching out and grabbing something. You can see that at this point in time, time is along the x-axis, and this figure shows you what's happening in the video at that point in time. So just as the hand is about to grab this block, you can see the activity of this neuron increases pretty dramatically. However, when the monkey watches a hand reaching out, opening up, and doing the same exact movement, notice that the action is exactly the same in both of these videos that the monkey was watching. The only difference here is the intention. Over here, the intention is clear. You're reaching out, grabbing the block. Over here, you're just reaching out and making kind of a funny shape with your hand. You'll notice that the cell all of a sudden doesn't like this. This neuron doesn't seem to be encoding the intention of this action, but rather the intention of reaching out and grabbing the block, despite the fact that the actual action is the same. But you could argue, well, maybe the neuron is responding to the presence of the block, or maybe the hand reaching out focuses the monkey's attention on the block, and so that this is really a block neuron or something like that. The experimenters tested this in kind of a clever way. So they show the monkey the same video, hand reaching out, grabbing a block, but the second half of the video was blocked out. Imagine putting a card in front of the video screen so that you couldn't see what the hand was reaching out and grabbing, indicated by the gray area here. This would be completely occluded in the actual experiment. So in this case, the monkey watched many examples of the hand reaching out, grabbing a block, reaching out, grabbing a block, and then it watched many examples of the hand reaching out and presumably grabbing a block, but you couldn't actually see it grabbing the block. But we think that the monkey probably inferred that that was the case based on having just seen this many times. And you could see that the neuron, the same neuron in this case, also likes this. The monkey seems to be inferring the same intentionality of this movement that it was inferring up here. However, on another day, they had the monkey watch this video many times. Hand reaching out, grabbing nothing, reaching out, grabbing nothing, reaching out, grabbing nothing, over and over again. And then watch this video. Hand reaching out, and we don't see the end of the action, but the monkey is probably inferring that it's reaching out and grabbing nothing the way it did dozens of times beforehand. And look at the activity here. Very little activity. Again, the monkey seems to be inferring the intention of this action. Now, importantly here, in both of these videos, what the monkey saw was identical. Hand reaching out, and then you don't see the end of the movement. Hand reaching out, and then you don't see the end of the movement. The only difference here is the inferred intention of the action. So Rizzolatti and his colleagues concluded that at least some of these mirror neurons may be important not just for understanding action itself, but the intentionality behind those actions. Okay. This one's going to really throw you for a loop, I think. The conscious decision to move and the movement itself occur at two different times. Now you may be saying, well, that's obvious. You decide what you're going to do and then you move. So the decision would have to precede the movement. It turns out that's not necessarily the case. Let me show you a now classic study conducted by Benjamin Libet and his colleagues. First, I need to tell you a little bit about the tools that they used in this experiment. There's something called a readiness potential. Remember that potential means voltage. This is actually an ERP, an event-related potential. So you record the readiness potential by recording the ongoing EEG over the person's motor cortex. And then you take a time lock average of the voltage leading up to a movement. So the subject is always doing some repeated movement, pressing a button, or in the case of the experiment I'm about to describe, moving their wrist. And then leading up to that movement, you see a change in voltage over the contralateral motor cortex, the motor cortex on the opposite side of the body from the hand or foot or whatever part of the body is performing the movement. This readiness potential starts about 500 milliseconds or more before the movement itself. This is about half a second. So you see a ramping up of voltage, change in the voltage over the motor cortex that's about to execute a movement in the contralateral hand. Now let me tell you about the experiment in which they try to learn something about the timing of people's conscious decisions to move. So in this study, they had subjects watch this dot moving around a kind of a clock. It would rotate all the way around in about 2.5 seconds. As it was rotating, the subject was told to just move their wrist whenever they felt the urge. Not to choose any particular number at which the dot was, but rather to just move when they felt like it, but make a mental note of where the dot was when they initiated the movement. Then they went and analyzed the data. They looked at when the movement occurred, they looked at when the conscious decision to move occurred, and they looked at when the readiness potential, this change in voltage, would start ramping up. Here you see it plotted along the clock. If this was where the light was when the wrist movement occurred, this is where it was when the subject reported the conscious decision to move. And this is where it was when it became clear that the brain was already preparing movement. This is plotting the same information in a different way. So this is showing you the ramping up of that readiness potential, that change in voltage over time. You can see the activity in the motor cortex starting to ramp up half a second or more before the actual movement. But the conscious decision to move didn't occur until later, until after the motor cortex was already starting to prepare for this movement. So who decided to move? This raises all sorts of interesting questions about the nature of consciousness, the nature of free will, the nature of choice. It suggests maybe that the conscious parts of our mind aren't necessarily in control of our behavior, at least not all the time. It's tempting to think the brain or the mind is being organized the way a company is. There's the conscious self way up at the top, which is kind of like the CEO of the company. And then you've got middle managers just below that. And then you've got all the lower level workers before that kind of executing the commands. Subjectively anyway, it seems like we're the CEO up at the top of the pecking order. In reality, it's probably better to conceive of the mind and the brain that creates it more as a committee. There are lots of different members of the committee going about their business, doing their different roles, performing their different functions within the organization. And they don't all communicate with each other constantly. And you can think of the conscious self as maybe being something like the PR person in the company who is communicating with the outside world. But the PR person may not always get complete information from what the rest of the governing committee is doing. And as a result, they may get the memo a little bit late after other parts of the brain have already started deciding to move. We're going to revisit this idea again when we get to the chapter on cognition and discuss split brain patients. These are people who've had their corpus callosum cut and essentially have two independently functioning hemispheres that are mostly unable to communicate with one another. You can set up demonstrations or experiments with these individuals and show that their left hemisphere can know things and do things that the left hemisphere doesn't. In most people, the left hemisphere is the one with language abilities, the one that can speak. But the right hemisphere can perform actions and do things without the left hemisphere knowing why. But nonetheless, the left hemisphere, the speaking hemisphere, often makes up reasons for why the right hemisphere has done what it's done. Because, of course, the left hemisphere doesn't see the right hemisphere as being separate from itself. It sees the body as being part of its whole, part of its self, its unitary self. Again, this implies that, at least in some circumstances, we become conscious of the decision to move after the process has already begun. And there's actually good evidence that this is the case not just for movements, but for other aspects of human psychology as well. There are plenty of things that go on outside conscious awareness that nonetheless influence our behavior and our choices. Next, let's talk about the cerebellum. Traditionally, this is associated with balance and coordination. The cerebellum is another important structure for movement. It's often associated with balance and coordination because those are the most obvious deficits that occur when one experiences damage to the cerebellum. People have a hard time making coordinated movement, and they become generally kind of clumsy. But the role of the cerebellum is a bit more complicated than just that. Its damage tends to cause trouble with really any kind of rapid movement that requires aiming or precise timing. For example, clapping hands, speaking, writing, and certainly things like throwing a ball would be impaired by damage to the cerebellum. People with cerebellar damage also seem to have trouble establishing new motor programs that allow the execution of a sequence of actions as a whole. The finger-to-nose test that you may have seen people with the police doing on the side of the road is another example of something that relies heavily on the cerebellum. If you ask people to close their eyes and touch their nose quickly, the first part of that movement is kind of a fast ballistic movement that requires very precise timing of the contraction of muscles to get the finger close to the nose. People with cerebellar damage, as well as people who've consumed too much alcohol, which we know impairs the cerebellum, often have trouble with this, and they'll either undershoot the movement, and then it takes them too long to use proprioception to get the finger right onto their nose, or they'll overshoot and smack their face with the finger. After functional brain imaging techniques gained widespread use, it started becoming clear that lots of different kinds of activities, lots of different kinds of tasks result in activation of the cerebellum. Things that would seem to have very little to do with balance, coordination, things that don't require any movement at all. So the cerebellum seems to play a role in other kinds of cognition. Cognition is a fancy word for thought. So tasks that involve only differences in memory or attention also often activate the cerebellum, which raises the question of what the cerebellum is doing then. If it's not just playing a role in the precise timing of movements, it may be that the cerebellum is important not just for the timing and sequencing of movements, but the timing and sequencing of activity in other parts of the brain as well. For example, there's work showing that people with cerebellar damage have difficulty just judging the timing of tones, or judging the timing of movements of objects on a screen. Things that don't require an overt movement. The next set of structures we're going to talk about in relation to movement are the basal ganglia, also called the basal nuclei. We saw these in the lab. They're a group of large subcortical gray matter structures in the forebrain that we know are important for the initiation of behaviors, movement, but also other more complex behaviors. And the basal ganglia are comprised of the following structures: the caudate nucleus, the putamen, and the globus pallidus. The first two, the caudate and the putamen, are together sometimes known as the dorsal striatum, or sometimes just the striatum, although there's also a ventral striatum. The basal ganglia play a role in lots of different aspects of human cognition and behavior, but if we had to summarize their role, we'd say that, for movement at least, their main role is to help select a movement by ceasing to inhibit it. You can imagine the basal ganglia hanging out down below the cortex, essentially telling the cortex no. Nope, sorry, don't move. Nope, nope. But occasionally permitting a movement by ceasing to inhibit the cortex. It turns out there are plenty of neurons in the basal ganglia that seem to encode the reward of stimuli that it's perceiving. In other words, actions that the animal or the person knows will produce an immediate positive outcome based on the situation. Those actions tend to be selected very quickly, and the movement that follows it tends to be more vigorous than it would otherwise. This is showing you kind of a simplified version of just part of the pathway by which the ganglia regulate the initiation of movements. So you can see that there's this area in the midbrain called the substantia nigra. It has axons that project to the putamen, and the green here indicates that they're excitatory. In other words, the substantia nigra has axons that extend out to cells in the putamen and release excitatory neurotransmitter there. In this case, it's the neurotransmitter dopamine. The putamen, in turn, inhibits the globus pallidus. The globus pallidus has axons that project into the thalamus that inhibit it. And then parts of the thalamus project to the cerebral cortex and excite it. As we'll see, this pathway is interrupted by Parkinson's disease. So Parkinson's is a neurological disorder characterized by muscle tremors, rigidity, slow movement, and especially difficulty initiating physical and eventually mental activity in the absence of stimuli to guide the action. In other words, self-initiated movement. We just learned that the basal ganglia encodes the reward value of an action. So external stimuli that help guide or initiate an activity tend to be pretty easy for people with Parkinson's disease. So for example, if I hold out my hand right in front of somebody with Parkinson's and say, touch it, they can usually reach out and touch it with their finger without much problem. But if I say to them, just stick your hand out right in front of you, it's almost the same exact movement, but they often have a very difficult time doing that, very difficult time initiating that movement. In the middle stages of the disease, they'll often have a difficult time beginning to walk. Once they've started walking, they don't have much of a problem continuing to walk, but initiating the onset of the walking can be difficult. They'll sometimes choose a target, like a spot on the floor right in front of them, and say to themselves, okay, I'm going to touch that spot with my right foot. And that helps initiate the walking movement pattern. Or they might pick a spot on the back of their thigh and slap it with their hand, again helping to initiate the walking pattern. Other symptoms of Parkinson's disease include depression, memory, and reasoning deficits. Most of the movement problems associated with Parkinson's disease are caused by a gradual and progressive death of neurons, especially in a part of the brain called the substantia nigra, which we saw in that last figure. It's in the midbrain, and it's relatively dark in color, hence the name black stuff. The substantia nigra has axons that release dopamine into the caudate nucleus and parts of the putamen as well. These tend to be excitatory synapses. So the loss of dopamine in the basal ganglia, as a result of the loss of the input from the substantia nigra, leads to less stimulation of the motor cortex and slower onset of movements. Let me show you how this works. So again, in the normal brain, substantia nigra is activating putamen, inhibiting globus pallidus, inhibiting thalamus, exciting cerebral cortex. In Parkinson's disease, though, you can see that the pathway by which the substantia nigra excites the putamen is compromised. These axons are dying off, and there's less dopamine released into the putamen. As a result, there's decreased inhibition from the putamen to the globus pallidus, so the globus pallidus is more active than it would be otherwise. That means its inhibitory input into the thalamus is exaggerated, so there's more inhibition of the thalamus, which means that there's less activity in the thalamus and therefore less excitation of the cortex coming from parts of the thalamus whose job it is to excite cortex and help initiate behaviors. So what causes this? There are different forms of Parkinson's disease. The early onset version of Parkinson's we know has a strong genetic link. This occurs in people under the age of 50. For example, Michael J. Fox has the early onset Parkinson's. And we know this has a strong genetic link, and we've identified most of the genes that play a role here. However, there's a late onset variety that affects people after 50, and the vast majority of people with Parkinson's have the late onset variety. In these folks, genetic factors play a relatively small role. There have been at least 20 different genes identified that do play a role, but each one contributes a very, very small increase in risk of Parkinson's disease. So if genes play a relatively small role in late onset Parkinson's, then there must be environmental factors that are playing a role. We don't really know what those are exactly, but there are a few things that seem to make a contribution. One likely candidate is exposure to pesticides and herbicides. People who live in rural and farming communities have a higher rate of late onset Parkinson's than do people who live in urban areas. And farm workers who are exposed to these kind of chemicals have an even higher risk. Head trauma also seems to increase the risk. Interestingly, tobacco and coffee use seem to decrease the risk. Using correlational studies of lifestyle factors that rely on surveys have shown that as tobacco use increases, the heavier people smoke, in other words, the less likely they are to get late onset Parkinson's. Now, nobody would recommend smoking as a way to improve your health. There are lots of other known risks to tobacco use that outweigh any potential benefits to Alzheimer's disease. But nonetheless, it's an interesting finding. Regular coffee consumption doesn't seem to carry a lot of health risks for healthy people. So go ahead and drink your latte, and maybe you'll help stave off Parkinson's disease. So how do we treat Parkinson's disease? The primary treatment for Parkinson's disease for decades now has been the drug L-dopa. L-dopa is actually a precursor for dopamine. But unlike dopamine, L-dopa can actually cross the blood-brain barrier. If you were to just take a dopamine pill, it would do nothing for your Parkinson's disease because the dopamine wouldn't make it into the brain. But L-dopa does. It's a precursor, which means that it's sort of the building block for dopamine. You have an enzyme that converts L-dopa into dopamine. When there's more L-dopa in the neurons that make dopamine, there's more of the substrate, more of the raw material for that enzyme to work on, and therefore you get more of the end product. However, it's often ineffective, especially for those in the late stages of the disease. It can be very helpful in the early to middle stages, but in the late stages there are just so few neurons coming from the substantia nigra to the basal ganglia that even if each of them was producing lots and lots of dopamine, it wouldn't be enough to effectively treat the symptoms. Also, the drug does not prevent continued loss of neurons. It really just treats the symptoms of the disease without treating the underlying cause of the problem, which is the neuron loss in substantia nigra. Also, L-dopa can enter brain cells throughout the brain. Any neuron that uses dopamine will end up having more of it as a result of having more of the precursor. This can produce unpleasant side effects, for example, nausea or repetitive movements, restlessness, sleep problems, sometimes even symptoms that are somewhat like obsessive-compulsive disorder. There are other potential treatments for Parkinson's as well. One that's been tried is the implantation of neurons from aborted fetuses. Obviously, this remains controversial, and it's only partially effective. It takes the neurons of several fetuses to have enough to do one implantation surgery. And most patients end up showing little or no benefit a year after the surgery. However, patients with only mild symptoms did show the benefit of failing to deteriorate further. This may be a result of the implanted neurons producing nerve growth factor or other neurotrophins that help keep the neurons around them from dying off. Stem cells are another possible treatment course. There's no stem cell treatment yet for Parkinson's disease, but pluripotent stem cells have the possibility of being grown in a culture and differentiating into any type of cell, including the cells in the substantia nigra. The hard part, of course, is getting them to integrate effectively and form circuits in the correct way. Another treatment that's become very widely used just in the last few years is deep brain stimulation. This is something like a pacemaker for the brain. It involves a pulse generator implanted underneath the skin of the chest and an extension that's run up underneath the skin through the neck and underneath the skin of the scalp to a lead that extends down into the brain with electrodes along the end of the lead, which then passes into the thalamus or the subthalamic nucleus and sends in continuous high-frequency pulses of current. In this video, this patient shows you the dramatic effects that this deep brain stimulation can have. First, he'll show you what his movement patterns are like with the stimulation turned on, and then he'll shut it off, and you'll see what happens.
This is me with the power on. I'm not making happy little faces for the video. These are tests that the neurologists make us do to test our reflexes. As you can see, they're wiggling, moving fine, and I've got very good movement. This is the remote control, and this checks that I'm on. It's on and okay. And this is me when I turn it off. You won't be able to read that, it's shaking too much. So it's almost an instantaneous reaction. I can't control this. It shakes some quality cocktails with the right. The left just looks like the royal wave. My voice is gone. It's harder for me to get get words out and speak. I'm completely rigid, and if I try to stand up, I fall. If anyone thinks I'm faking this, I'm not. I can't control the tremors and dyskinesia. The twisting in my neck is called dystonia and it is really quite uncomfortable. So, as you can see, the tremors are getting much worse and I'm in quite a bit of discomfort, and I'm about to drop the remote control, which will be fun because I won't be able to turn myself back on. So I think party time is over. Almost automatically. The power comes back on. I'm steady.
Okay, so you can see how effective that is. It used to be that the surgery was reserved for people in very late stages of the disease, but now it's often being used in younger patients and people with less severe Parkinson's disease because it's been shown to be safe and effective in so many others. Another movement disorder that we're going to talk about is Huntington's disease. Huntington's is characterized by debilitating motor and cognitive symptoms. It affects about 1 in 10,000 people in the United States. You usually don't start seeing symptoms until between the ages of 30 and 50. The vast majority of people who get Huntington's start seeing symptoms during that age range. The disorder is associated with gradual and extensive brain damage, especially in the caudate nucleus, the putamen, the globus pallidus, but eventually in parts of cerebral cortex. So in other words, the parts of the brain that are hit hardest are the basal ganglia. This is showing a coronal section of a healthy brain and a similar section in an individual with Huntington's disease. You can see the loss of neurons, the degeneration, the enlargement of the ventricles. In some ways, you can think of Huntington's disease as being kind of the anti-Parkinson's. In Parkinson's, there's too little excitation coming from the substantia nigra, which results in too little excitation of cortex. In Huntington's disease, damage to the basal ganglia, primarily the globus pallidus, results in too little inhibition of thalamus, which means too much excitation of thalamus, and therefore too much excitation of cortex. Instead of having difficulty initiating movements, as Parkinson's patients do, these individuals have a hard time not initiating movement. They have excess movement that they can't control. Initially, the motor symptoms include arm jerks and facial twitches, but eventually these progress to tremors and writhing that affect walking, speech, and other voluntary movements. These are often referred to as choreic movements, or Huntington's chorea, not chorea like the country, but chorea like choreograph, because the movement looks almost like a kind of dance. There are psychological symptoms that go along with this as well: depression, memory impairment, anxiety, hallucinations, delusions, poor judgment, alcoholism, drug abuse, and sexual disorders. Many of these, for example the sexual disorders and drug abuse, can be seen as kind of an extension of the movement disorder. Just as these patients have difficulty holding back the impulse to move, they also have difficulty holding back the impulses to do other things that they probably shouldn't be doing. There are presymptomatic tests for Huntington's disease that can identify with high accuracy who will develop the disease and even roughly how old they'll be when they develop the disease. What do you think? Would you have the test? The disease is controlled by an autosomal dominant gene on chromosome number 4. So if you think that through, it's autosomal, so it's not on one of the sex chromosomes. It occurs equally in men and women as a result of that. It's a dominant gene. Most disorders are a result of recessive genes because when there's a mutation, typically you lose the function of that gene or the protein produced by that gene, and so you need 2 bad copies in order to lose the protein altogether. In this case, the gene produces a bad protein, and all you need is one copy of that gene in order to have the bad protein. So how does it get passed on? If you think this through, this means that an individual with Huntington's disease typically only has one copy, and so each of their children would have a 50-50 shot at getting Huntington's disease themselves. Huntington's is one of several disorders associated with consecutive repeats of the combination CAG. These are 3 base pairs that keep repeating within the gene. These base pairs code for the amino acid glutamine. So the resulting protein ends up with sort of a long tail of glutamine amino acids attached to it. The higher the number of repeats, and therefore the higher the number of glutamine amino acids attached to the protein, the more certain and the earlier the person is to develop the disease. We all have quite a few of these CAG repeats at the end of this gene and other genes. If you have less than 35, you're pretty safe. But the more you get above 35, the more likely you are to get Huntington's disease, and the earlier you'll get it, and the faster the symptoms will develop. Unfortunately, there's no effective treatment for controlling symptoms or even slowing the course of the disease currently. Although because we've now identified the protein produced by this gene, there are several potential drugs that are being tested that may help treat the symptoms by counteracting that protein. This slide here is showing you the relationship between the number of CAG repeats, and therefore the number of glutamine molecules in the tail, and the age at the onset of symptoms. You can see individuals with 40 or so of these, on average, get the disorder in their early 50s. The main thing to take away from this is that as you get more and more repeats, the age of onset gets earlier and earlier, until by the time you have 80 or so repeats, you're getting it in your teenage years.